This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 152, recorded October 6th, 2011. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from Western Massachusetts is Alan Dove.
Good to be here.
Welcome back.
Thank you.
And do you have beautiful blue skies and sunny days here?
Yes, yeah. It's a gorgeous New England autumn day, just crisp enough to feel just right.
Yeah, pretty nice. Mid-70s here.
Yeah.
Beautiful. But the Hudson River is very brown. Which is
there's been a whole lot of rain lately.
Yeah, it's runoff from upstream. Also joining us today from north central Florida, Rich Condit.
Hi, everybody. How you guys doing?
All right.
Pretty well. Yeah, when the sun is shining, I feel better.
Good. Yes, I've noticed that phenomenon. You know,
I don't know why.
Well, it happens.
And when it's cloudy, I feel like oppressed or something like that.
Oh, we're a diurnal species. We like the light.
Yeah, we are diurnals. And that's it for today. There's no one else, in other words. Before we get started, I want to say goodbye, Steve Jobs.
Yeah.
I used a lot of his computers in my lifetime.
Rest in peace. Yeah, my first computer was a Mac Plus.
Yeah,
cool. My first computer was an Apple II Plus when I was in middle school.
Yeah, I had a Mac Plus. It was my first computer.
Matter of fact, I still have the Mac Plus. It's in my son's closet. He still hasn't. Are you listening, Jeremy? He still hasn't cleaned out his room, you know.
Well, you should keep that Mac Plus because they.
Oh yeah, that's he's got a whole string of Macs.
Yeah, collectors. Okay. The inside of that first one had their signatures in it.
He had this wonderful. My son had this wonderful post on his Facebook page this morning that was, you know, the old Mac icon.
Yeah,
it looks like a Mac Plus with a smiley face.
Yeah,
it was the same thing with a sad face.
No,
it was really cute.
Some site reskinned their whole site in the style of the old Finder OS, you know, with the lines and that sort of thing.
Personally, I thought his first product was pretty amazing too. The Apple II Plus, the Apple II series.
It was cool.
That was the dawn of personal computing.
Yeah, actually, we had one of those in the lab. It was my first own personal computer that was the Mac Plus, but we had Apples in the lab before that. You know, and I was talking to somebody about this morning. When this first came out, I just didn't get the whole concept of having a computer. Like, what do I need a computer for? I thought of them as things that you did math with, and I wasn't going to do any math for sure, right?
Yeah.
Now.
Yeah, but once you played your first game of Little Brick Out, you just
Zork.
Yeah, Zork. Anybody else do the Scott Adams adventure games?
Mm-mm. No, I didn't.
Oh, I wasted many, many hours of my youth on that.
Did other ones. It's amazing how much press he's getting, Times.
Yeah, well, he really was a visionary.
Yeah, it's pretty impressive. I used to enjoy watching his keynotes. They were always interesting.
Yeah.
Good presenter. Well, we'll see what happens to Apple. You know, there's going to be some inertia, but who knows? You don't find a guy like that every day. Anyway, some of our picks are related to that today. Today we have two papers. One of them just came out in the journal called MBio. This is an open access journal of the American Society for Microbiology. And it's called Raw Sewage Harbors Diverse Viral Populations. And the first author is Paul Cantalupo. The last author is James Pippas, whom I know. And one of the authors in the middle there is Roger Hendricks, who was on TWiV in New Orleans. And if you listen to that one, you know that he's interested in characterizing the viruses that are out there.
Mm-hmm.
I love the first sentence of this paper. Viruses are everywhere.
Right.
Yes.
How big is the viral universe and how many types of viruses exist? And that's the question here being addressed.
On Earth, every species of bacteria, archaea, fungi, plants, worms, insects, and animals likely to harbor numerous viruses. The implication is that Earth is just one place we're looking.
Yeah.
How about other planets?
Oh, that's a good point.
Well, I suspect if we find life elsewhere, we will find viruses elsewhere.
Yeah, maybe. Depends. So they note that we know about 3,000 different viruses at this time. But of course, massive sequencing technology allows you to just sequence the virome of different environments. And here, they picked raw sewage. And I love this. Raw sewage represents the effluence of society. Is that a correct construction, Alan? Effluence?
I'm not sure. I guess that's the correct use.
Yeah. Let me see. Effluence.
Yeah, it's a substance flowing out from something.
Yeah, I guess it flows out of society, right?
Yeah.
That's very graphic, isn't it?
It certainly is.
Sewage is very easy to get. Raw sewage, unprocessed, I suppose.
There's lots of it.
They're giving it away.
So this is the stuff that comes out of sewers, right? So whatever's in our guts, whatever we eat, passes into it. And they also note that I guess sewage has other stuff. It's got not only human waste, but animal wastes also.
Sure.
Where would that come from? Just the environment?
Cleaning out the litter box.
You flush it down the toilet?
Yeah.
Wow.
Not the litter, but not to get too graphic, but you've got the clumps of the urine, and those go in the trash, and then you have the little
I see.
The number 2, and that goes in the toilet.
Also, plant material, which gets
probably
into the system.
runoff. I'll bet you runoff contributes a lot of animal detritus.
Gets into the
and that's also a good point, because the cities that they looked at are probably all using what are called CSOs, combined sewer outfalls, where
that?
you have the rainfall and the sewage going into the same system.
I see.
So everything that washes off the streets, New York is like this. Everything that washes off the streets and goes down those drain grates, and everything that comes down the toilets gets combined and sent through a processing plant.
I see. So that when it rains heavily, that's why they overflow?
That's why they overflow, because the rain overloads the system, and then you get some raw sewage flowing out into the waterways, which is a major criticism of that type of system. But as you can imagine, it's not the kind of thing that's really easy to fix.
So both you, Alan, and Dixon seem to know an awful lot about sewage.
Well, Dixon's a parasitologist, and I'm a writer.
Yeah, Alan knows about everything.
I produce a lot of
oh, I see. That was good.
Yeah, it's
so
something that comes up pretty often in the stuff that I cover.
Could be.
Well, I think it's actually fascinating to consider what happens in a big city to all of the stuff we flush down a toilet. There's a lot of pipes going somewhere.
And in fact, one of the stories I did a few years ago, I was I won't say that I was the first on this, but I was one of the first reporters on this pharmaceutical pollution problem, which you've probably heard about more in recent years, where you take some Tylenol because you have a headache, and then a little while later you go to the bathroom. Well, a lot of that Tylenol actually washes out of your system.
Yeah, sure.
So you can detect now with the new methods that are available, you can detect various prescription and non-prescription drugs flowing out in the sewage, and in places that have small aquifers that they're draining into. This can build up to maybe worrying concentration. So there's the potential that this could do some damage, but nobody's quite sure. Some of these are compounds that are long-lived in the environment.
Yeah, we did a TWiV a few years ago on Tamiflu in Japanese river water.
Right.
Which comes from that source. People excreting it. Dixon would say here in New York City, if you took the fecal material from these sewage treatment plants and dried it, you could actually generate a lot of energy from burning it.
Yeah.
Okay.
And you can clean it up to a certain extent so it wouldn't be offensively smelling, I guess. Or
So when they're talking about raw sewage in these experiments, this is stuff that is coming into the sewage treatment plants that they have done absolutely nothing to. Is that right?
Right. It has not been treated.
Okay.
So is this what you would call a suspension? It's not a solution, right? It's
Right,
heterogeneous.
It's probably a suspension.
Okay,
Yes.
with bits of stuff floating. And it probably smells really awful, right?
Yeah.
I'm sure.
Okay,
Right.
so they collected this from, in big volumes, 10 liters,
Yep.
Pittsburgh, Barcelona, and Addis Ababa. Now,
I'm sorry. They pooled them.
yeah, they pooled them. I
I'm
know.
really
I was really bummed about that.
Yes.
Yeah, I wanted to know the breakdown.
Yeah.
Now, they picked Pittsburgh because two of the guys, that's Pepys and Hendricks and some of the other authors, are from there.
Right.
I don't know about Barcelona.
So they're at Pitt looking at toilets.
Now, there's a University of Barcelona guy, so that explains that. And Ethiopia, that I don't know. So it must have a connection, right?
Yeah.
All right, so they take this stuff. They actually look at it by EM. You can see a lot of particles in it.
They must have these still separated out somewhere. I can't believe that they won't report the distribution in the individual things. I mean, it's just...
It would be criminal.
Yeah.
Yeah.
After
Well,
all
maybe
this
they do.
stuff.
Maybe they do.
That's
yeah,
another
it may
paper.
just be that for this first one, it's it was they wanted to get it done, and so what do? You pull your samples and sequence and see what's there.
So they
All right.
took the stuff. You could see by EM there are particles. Most of them look phage-like, but there are other spherical ones. And then they say they concentrate and purify it by organic flocculation and DNase treatment. Anybody
Right.
ever do organic flocculation?
The closest I've done to that, and I don't think it's actually flocculation, because I think it's really a precipitation, is a PEG precipitation,
Yeah,
polyethylene glycol. But that's not flocculation, right?
So
At
what
least
does,
I don't
what does
think so.
flocculation do? Just precipitate small stuff?
I think so. And I looked up the reference that they have for this, and it was actually developed for seawater. It's Barcelona. I think this is maybe one of these authors is on this same paper. So they developed it to precipitate viruses from seawater, and they just take milk proteins, I guess, your basic non-fat dry milk that you use for things like blocking westerns, and toss it in there, and I guess the stuff sticks together, and then you precipitate it out.
Yeah, apparently particles below a certain size will stay in suspension just by basically Brownian motion, I guess.
So the viruses stay behind. Is that the idea?
No, I think that this actually aggregates and precipitates them.
Yeah, I think the
Okay.
viruses come down.
They would stay in suspension otherwise, but if you add a lot of this stuff, this milk protein and stuff, it forms aggregates with small particles and then settles.
Yeah.
That's my understanding.
So,
And just backing up a tiny bit, I just noticed in Figure 3, they do actually deconvolute the data partly by location. They've
Okay.
color-coded this figure. They've got the different viral families split out, and they've color-coded the figure by which city is being represented in it.
Okay.
So the blue,
okay.
green, red dots in there are representing the different city samples. They actually look pretty close, which is not that surprising. People are people.
So it sounds like they pulled the results in silico to present them in the paper, maybe.
Maybe.
Well, let's see when we get to that. Let's see if we could sort it out. I didn't notice that it was separated by.
So in Wiki,
Location.
it says that flocculation is a process where colloids, and viruses in this case would be colloids, come out of suspension in the form of flock or flakes by the addition of a clarifying agent. Differs from precipitation in that prior to flocculation, colloids are merely suspended in a liquid, not actually dissolved in a solution.
Right.
Oh, that's interesting. So I wonder if PEG precipitation is actually flocculation. I looked up a number of the flocculating agents, and polyethylene glycol was not one of them. But
Right,
at any rate, it makes all these things aggregate, and it settles out.
So they basically are adding a skim milk solution to the 10 liters of raw sewage. They let it mix, and then the flocculant's allowed to settle in centrifuge.
Right.
So you're not doing any kind of... I mean, what happens to the bacteria, the fiber from the feces?
So it's going to be a whole bunch of... Well, I looked to see whether there was any sort of process that they did before the flocculation to get out the really big stuff. And
right.
It's not written in there, but I can't... there must be all sorts of solids in this.
There is.
And it must have been clarified before they flocculated it. Certainly I would do that. Otherwise, you can have all kinds of junk.
Here's what they do. They flocculate it. They say they take the sewage, raw sewage, acidified raw sewage, flocculate it. The flocculants are allowed to settle. Centrifuge. The concentrate is resuspended in buffer in eluted and pH 9.5 buffer. Then the suspended solids are separated by low-speed centrifugation.
Okay.
And virions in the soup are concentrated by ultracentrifugation.
Got it.
So they're getting rid of some stuff.
So that's where they get rid of the big stuff.
Right.
No.
They're not doing a filtration, but they're doing a subsequent centrifugation that's going to bring down the really big stuff.
Right.
And then the virus,
And the virus. And I gather this flocculation method enriches for virus, so it's not going to eliminate the bacteria. But as we've talked about before, even filtration wouldn't do that. And doing a filtration on these kinds of volumes would be kind of painful.
Yeah, painful, and also it's dirty, so it's going to clog your filter.
Yeah.
And then they have a nuclease step in there.
Yeah,
Okay?
they have a DNase treatment,
Right.
Just DNase treatment. They don't ribonuclease treat it. I don't think it's just DNase treatment.
Yeah, I guess DNA is...
But of course, if you use micrococcal nuclease or something like that, that'll get rid of RNA as well.
Mm-hmm. DNase. Let's see which DNase they used. The RNA is probably... is there a lot of RNA in sewage, you think?
I don't know.
I don't know.
I don't know how stable it is. They just say DNase. They call it DNase, catalog number EN0523. It's just DNase. All right, then they extract total DNA or total nucleic acids and reverse transcribe it and deep sequence it.
Right.
So you'll get basically DNA, which won't be affected by the RT, and RNA will be reverse transcribed into DNA. So you get both DNA and RNA viruses.
Mm-hmm.
So they got almost 900,000 reads, 278 million bases from all three samples. And then you give it to the bioinformatics folks.
Ask them what's in it.
I say, what's this? What are all these ACTGs? 8,491 reads were most closely related to eukaryotic viruses, 37,000 bacteriophages, 27% were identified as bacterial. Most sequences, 596,000, showed no sequence relationship to any known sequences in the database.
Of course.
And are most likely to be derived from novel viruses.
Well, that's where I have an issue.
Okay.
Because we went through this in a recent episode where there was, in response to an email, somebody wrote in and was wondering about a paper that it was looking at the virome of the gut of
pigs.
Some animal, right?
Yeah,
what
pigs.
was it?
Yeah, that was pigs. Eric Delwart's study.
Yeah, they had a lot of. Even though they actually filtered that stuff and nuclease treated it, there were a number of eukaryotic sequences around and that kind of stuff. And the question was, where does all that come from? And Eric, who in my mind is still famous for his quote, DNA is everywhere, it's really a problem, right? Said that they figure that there's a lot of aggregates of protein and nucleic acid, not to mention ribosomes, but those would be recognizable, but that make it through these treatments. So I think just because now maybe they've done something else here to either bioinformatically or otherwise screen this, but I couldn't see it. I don't think you can assume that just because you can't identify it that having come through this procedure, it's viral.
Right. Just because you're a virologist doesn't mean you found a virus.
So you think that some of these might be unknown bacteria that we haven't seen before?
I suppose, or
fungi.
Right. Bits of DNA from organisms that we haven't sequenced yet. You know, this is Eric's dark matter, right?
Yes, biological dark matter.
Yeah.
I mean, that doesn't mean there aren't a lot of unknown viruses in there. I don't mean to take anything away from the paper, but I don't think you can assume that they're viruses just because of the way the treatment was.
I mean, I'm sure some of those. I'm sure some proportion of those are viruses. It would be weird if they weren't. But we can't know, since they don't match any known sequence. We can't know whether they're viruses.
Yeah. Of the total of 900,000 reads, over almost 600,000 had nothing in the database. It's a lot.
Right. Yeah. That's what Eric called dark matter.
Dark matter. All right. They found 234 known viruses from a variety of different families. Double-stranded DNA, single-stranded DNA, plus sense RNA, and double-stranded RNA.
No negative sense RNA.
Yeah, that was odd.
What's up with that? I mean, there's a lot of negative sense RNA viruses out there.
There certainly are. Don't get it, because they got both enveloped and non-enveloped viruses, so that's not it. That's biasing against negative strand viruses. I don't know. It's a very good question. And they don't actually mention it, right?
No, they don't address that, though. You know,
would their RT have discriminated?
They're probably using random primers.
Actually, you know what?
So it shouldn't.
I wonder if most of if very few of the negative strand RNA viruses are enteric viruses. I've never thought about this. But if you look at the list of guys they found, adenovirus, astrovirus, Norwalk, papilloma doesn't really fit. But and I don't know about AAV, but some of these others, I wonder if it's biased towards things that you might find in feces or urine and against things that you might not.
You know, they could be very low abundance, and they're not picking it up because we know that's an issue here.
Mm-hmm. Yeah.
I know flu will pass through the intestine. Certain strains will pass through, even though they don't replicate there. They will pass through, but it may be too low to detect by direct sequencing.
At any rate, it's interesting. There's a distinct bias against negative strand RNA viruses.
Yeah, either they're not abundant, or there's some kind of systematic problem in the assay that's failing to detect them.
They also say, like other biomes that have been studied, the virome of raw sewage is dominated by bacteriophages. 80% of the matched sequences, bacteriophages, and also a lot of them, most of the eukaryotic viruses, 90% are from plant viruses.
Yeah.
So
they also found this non-A, non-B hepatitis virus.
Yes.
Yeah.
Turns out to be a bacteriophage, right?
Right.
So this is a virus associated with non-A, non-B hepatitis.
Right.
Not proven to cause it. And now we see that it is a phage, so it doesn't cause non-A, non-B hepatitis, right?
Right. Because it was originally isolated from stool from hepatitis patients.
Yeah. It's very cool.
Yeah. So just by doing this fishing expedition, they've now found out something really interesting about this virus.
Yep. That's cool. Now,
I think that this is. Pardon me, I'm going back here. I'm just looking over at my old table where I take for the medical students. I take the virus families and I give representatives of human viruses, and I don't see any, frankly, enteric viruses amongst the negative strand RNA viruses, whereas they do exist among the positive strand RNA viruses. Obviously, a double-strand RNA virus is reovirus, rotavirus. And the DNA viruses. So there are good examples of enteric viruses in the other categories, but not obviously in the negative sense RNA viruses. So it may be biased by the selection of sewage.
Yeah, but it could be that they don't replicate in the gut, so that's why we're not seeing them.
Right.
And that in itself is interesting. Why don't negative strand viruses replicate there?
Okay, sorry.
Among the viruses, human viruses, adenoviruses, astroviruses, the Calices, Norwalk, Sapporo, human papilloma 112.
I don't know what that's doing there.
Yeah.
Of course, I don't know what 112 is either. I don't know that. I can't think of examples of papillomaviruses that are enteric, though. Could be
skin.
Yeah, why
I think
not?
they mentioned skin could be sloughing off.
Yeah.
Parvoviruses, bocaviruses, and of the picornas, they have Aichi and Parecos, but no polio, no enteros of any other kinds, which is surprising to me.
What are these guys, Aichi and Pareco?
Yeah, these are recently. Well, Aichi is a recently described virus associated with gastroenteritis. Same with classivirus. The Parecos used to be echoviruses, and you would expect them, but there's no enteros at all. It's very interesting.
Yeah.
Yeah.
And then a single polyoma virus.
Right. And they comment on the limits of their sensitivity relative to the polyomavirus because they expected to see the common human polyomavirus is either JC or BK that hang out in our kidneys all the time and show up in the urine. So they actually went specifically looking for those by PCR. And they're there in the sewage, but they weren't picked up by the deep sequencing. So that they use that that's a nice sort of internal control to say, we know we're not getting everything.
Right. The the net has holes in it.
Right.
Yeah,
I
was also interested to see this negative control that they used in one of their PCR reactions. They explain in Figure 5, Shinola was used as a negative control, right there in the figure legend.
Very important.
Yeah, so they mention in the acknowledgments, we thank Justin Brown and W. Mark Peepus for providing Shinola.
It's an appropriate control for sewage, right?
It certainly is. Yes.
Absolutely.
And it's good to know they can tell the difference.
And we'll put a link to that so everyone knows what we're talking about here.
Yes.
Very clever guys.
Yeah.
But what really was the control? Well, there isn't any control. I mean, this is an observational study. You don't...
Right.
Right.
Are you going to... Let's think of what a control would be.
Yeah, what's your negative control for sewage? Which I was
interested actually to see in this list of viruses that showed up adeno-associated virus. Okay, because I know it's out there, but I don't think I've ever seen that show up anywhere but in cell culture.
Mm-hmm. They spend the rest of the paper discussing their approaches, their bioinformatic approaches, to demonstrate basically that known viruses represent a small fraction of the viral universe. Most of the sequences that they have that are unknown, they seem to be viral, and we don't know what they are, which is very interesting. So they conclude these observations emphasize the vastness of viral diversity among different biomes.
Very interesting.
Very cool study, and just the first one. There'll be many more. This is actually not that expensive a study any longer.
No.
So you can do it.
And it's getting even cheaper.
It's the bioinformatics that is getting to be a real issue.
Yeah.
It's cheap to do sequencing, but then what the heck does it mean?
Yeah.
If you consider that half the data they collected is dark matter, and probably half the data that anybody collects is dark matter nowadays in these sort of discovery type investigations, it's going to take tremendous effort to sort through that and figure out what it is.
Yep.
They also say that they believe they're underestimating the number of viruses present for a variety of reasons. And this is actually useful in the case of raw sewage. So the origin of viruses is human and animal feces and urine, plant material from domestic and agricultural areas, as well as from insects and rodents that are in the sewer system. In their growth, the sewer can grow out bacteria, rotifers, amoeba, fungi, and they become infected. It's really a complicated ecosystem.
There was a huge fraction of plant viruses.
Yeah.
It was like, as I recall, like half of them or something.
Well, of the eukaryotic viruses, 90% of the reads, of the known reads, were plants, but of course then there are all the unknown ones. All right, now here's something that I don't know if you know about this. They make a statement at the beginning of the discussion, which I'm very surprised is present here. It says, the International Union for Conservation of Nature lists nearly 1.8 million species of living organisms on Earth. Now, about a month ago, the Washington Post covered a story, which was published, I think, in PLOS Biology, where they estimated that there are 8.7 million species on Earth, and they estimated the bacteria at, I think, 10,000 species, and archaea at 1. And ASM freaked out over this because a teaspoon of soil contains 10,000 different bacterial species.
Weird.
So they ignored the bacteria, and this seems to also ignore the bacteria.
Uh-huh.
Yeah.
So I'm surprised, coming from an ASM journal, that this came through.
All right.
I don't know what that's about.
Well, they needed to cite somebody with an estimate, and the Union for Conservation of Nature, I mean, traditionally this stuff has been about the charismatic megafauna and
right.
the insects, and that's about it.
They ignore the bacteria.
Stuff with legs and flowers, right?
Yeah, stuff you can see.
But clearly the bacteria, the microbes, need to be counted.
Well, and the fungi, and there's good evidence that we've massively underestimated the diversity of the fungi. You know, half of them probably are undiscovered, and then bacteria, as you say, you know, everything we can't culture we haven't discovered, and viruses on top of that. So
yeah, and finally, it's worth emphasizing. They say, you know, the exploration of the viral universe has only just begun. We have to look at wastewater all over the world at different times. You know, I mean, can you imagine the data that's going to be generated? And that's just for sewage. I mean, you know, people themselves, our gut microbiomes and viromes.
I kind of expect the sewage is going to show some uniformity. There'll be variability from place to place, but it's all people and it's human societies producing sewage. But it'll be interesting to see how much it varies.
But here's the thing. All right, at some point, you'll explore most of it. You'll get 90% of the species. But what does it mean?
There is that.
All right, so I know there are a lot of viruses out there, but what does that mean? And now that's the hard question. This is low-hanging fruit.
Right.
Yeah.
Right.
That's why you have trouble getting discovery-driven science sometimes through the study sections, because that's the question. What does it mean?
Right.
Yeah, I mean, I think this should be done. I'm not saying not to do it, but at some point you can't look anymore, right?
Right.
You got to start doing experiments, and
right.
Maybe you look in an environment that has seasonal changes, and you look at the virome changes and try and figure out what that means, that sort of thing. But it's cool.
I think of it as kind of the way back in the 19th century there were all these scientific expeditions to various places, and it had finally gotten to the point in the late 1800s that people could actually get to inaccessible areas of the world and deep into rainforests and into the Arctic and all these places, and you had this massive series of expeditions sponsored by museums and universities, and they were just going and collecting all kinds of stuff.
Yeah.
And the result was just basements and warehouses full of these collections, many of which have only been just slightly analyzed. And now people are finally starting to go back to some of that stuff and discovering new dinosaurs in the basement of the American Museum of Natural History or the California Academy of Sciences. And this stuff's just been sitting around since whoever went and got it in 1890, whatever, or the early 1900s. And I think right now we're at that point in microbiology and in virology where there's just all this exploration has suddenly opened up because of the technology. We can get to these places we couldn't get to before. We can access genomes that were completely inaccessible because we couldn't culture the organisms and we couldn't sequence them. And I think a lot of these data are just going to be warehoused, hopefully in some format that we'll be able to access 20 or 30 years from now. And we'll be mining them for years.
Yeah.
Well, that's going to be the key, being able to mine them, because once they're thrown up on a server, it's lost unless you know how to look for it.
Yeah, and as I say, I hope it'll be archived in some way that we'll be able to access, and these don't all just kind of fall by the wayside.
Well, it's like there needs to be one big dark matter server somewhere, so that so that when anybody goes and sequences some new organism somewhere, one of the things they can mine is the dark matter, and when they've identified that as belonging to an organism, they can subtract that stuff from the dark matter.
Sure.
Well,
Right.
that's what these database projects are hoping to do, and to some extent they're linking themselves together. But there's not there should probably be some more standardization of that.
Right.
Well, speaking of large amounts of data,
Yes.
that's a good segue
Good one.
to our first sponsor. It's an old friend of ours who's come back for another round, and that would be Drobo, the makers of various storage solutions. And they've decided to give away a free Drobo to one of our listeners. This is a 1-week-only contest. I want to see how deep the TWiV audience actually goes. So here's how you do it. You can go to Twitter and post a tweet there. Or you could do it at facebook.com/thisweekinvirology. Just a short tweet or note on why you should get a free Drobo. Why do you want one? What are you going to do with it? Okay, so either Twitter or facebook.com/thisweekinvirology. If you do it on Twitter, use the hashtag #TWiVDrobo, all one word, so we can find them. And then the last thing you need to do is fill out a form, bit.ly/DroboTWiV. Okay, bit.ly/DroboTWiV, one word. You fill out a little survey there, and then you're entered in the contest, and after a week, we'll pick someone out. So you can get a free Drobo.
And they are very, very, very handy devices.
Do you have one now?
Yes, I do. Back when they were sponsoring us before, they offered a discount, if you'll recall, and I took advantage of that.
Yeah, I have quite a few of them here in the office and at home. They're very useful for backing up data.
Backing up large quantities of data.
Large quantities, such as you might generate in a sequencing project, such as the one we just talked about.
Or just pictures of your kids.
Or pictures or whatever you got there. Check them out, drobo.com, and enter our contest. 1 week you have to do this. And it's only for people that are going to be listening, because we're not posting this on the website, so you have to listen to TWiV to pick this one up.
Yeah, and do this so Drobo will give us lots of money.
That's right.
Yes.
If it works out well, they'll come back. So we thank Drobo for their interest again in supporting TWiV. Our next paper is a wonderfully short one. It's called a Brevia in Science, one page.
I love this format because they're usually it's got to be something cool that you can explain in one page of science.
Yeah, and of course it does have supplemental material.
Sure.
But it's a nice page, so if you want to read something quickly. So this is called A Gene for an Extended Phenotype. First author is Kelly Hoover. The last author is James Slavicek. And this actually relates to something we discussed maybe last episode or the episode before. The idea that viruses can change behavior. And I think David Esteban had asked if we knew of any examples of that.
Yeah, this is sort of the beginning of the whole zombie thing, right?
Right.
Yeah, and then someone sent us an email, which we read either last time or the time before, and she said, I know this baculovirus, which makes caterpillars crawl higher up in the tree, and then they burst, and the virus rains down on the lower leaves. She said, I don't know where I saw that, but that's what I heard. And now the paper comes out on this, which is very cool. So extended phenotype is a phrase coined by Richard Dawkins to indicate when a gene can influence things that happen beyond where the gene is. So like in another organism, right? So that's what's going on here, and you'll see what that means. So is this a cool paper?
This is neat.
Yes, it's very cool.
So basically the baculoviruses, these are big DNA viruses that infect various insects, right, Rich? Mm-hmm. Are you an expert on baculoviruses?
I know a thing or two about baculoviruses. I get baculovirus papers to review now and then, and I have some baculovirus buddies, Plus, there are insect pox viruses that do this kind of thing. There's always been sort of some caterpillar theme in my life. When I was a graduate student, there was a postdoc in Joan's lab that was working on fibroin, the silk protein, and she reared bombyx in the laboratory. And then here they had a guy working on insect pox viruses. What's fascinating is that these viruses that infect caterpillars do indeed just liquefy these creatures. It's disgusting, right?
So it's something you've studied a little bit. You have a baculoground in it?
Good, thanks. Yes. Thanks, Alan.
Baculoground, very good.
Baculoground.
So this behavior where the baculos induce climbing behavior in caterpillars is apparently old. It's known for 100 years. It's called
Wipfelkrankheit.
You guys, good. You guys speak German?
No.
I got a little German in there.
Do it again, Rich.
Wipfelkrankheit.
What does that mean? Do you know?
No relation to Walter.
Krankheit is sickness.
Okay.
But Wipfel apparently is treetop.
Treetop disease. Okay.
Right, because it's treetop disease. And they actually give a reference to this, O. Hoffman, and it's got some
German name.
Fancy German name, 1891.
Wow.
All right. So people knew that when these caterpillars got infected with the virus, they climb up to the top of the tree.
Yeah.
Well, they didn't know. 1891. They didn't even know they were being infected by a virus. They probably knew they were sick because they climb up to the top of the tree and melt.
Treetop disease. That's what they knew. Yeah. So they looked at this in this paper in a baculovirus that infects the gypsy moth. All right, L. dispar is the gypsy moth.
Have you ever been around for one of the gypsy moth invasions?
Yeah, I have.
Oh, yes.
Oh,
yeah.
We had one in Maryland when I was, I guess, when I was about in high school, and it was bad.
That was probably the same one when I was in graduate school, and it just stripped the trees in Connecticut. Just absolutely stripped everything.
Denuded. It looked like wintertime in August.
You could hear them. I'm not sure if I was hearing the crunching or the pooping.
Or the frass. Yeah, and the frass was on everything. It just coated the car every morning like black snow. It was awful.
Amazing.
So the infection causes, this in gypsy moth, this virus, the baculovirus, Limantria dispar, nucleopolyhedrosis virus, causes natural epizootics in the gypsy moth. Before death, the larvae, which are infected, climb to the top of the trees to die, liquefy, and release millions of infected virus particles with dispersal facilitated by rainfall. So it sounds like a dispersal mechanism induced by the virus, right?
Mm-hmm.
Right.
So the healthy guys, which are not infected, hide in bark crevices or climb back down the tree to the soil during the day to avoid predation. And then at night they go back out on the leaves. So the infection induces this weird behavior. Mm-hmm. And they want to know why. So
how.
Actually, how. Dixon always tells me don't ask why in biology. Yes, ask how. Correct. And they have a cool hypothesis that the disease is induced by a viral gene that antagonizes 20-hydroxyecdysone. Now, I bet Alan knows what 20-hydroxyecdysone is.
That induces molting, I think.
Yeah. And what's molting?
Molting is that's how the caterpillar or the insect grows. So it sheds its old skin and grows into its new one.
Yeah, molting. So it's stimulated by this hormone, which is produced by the caterpillar, right? It has a certain timing in its life. It does this. And so the idea is that this viral gene, it's called ecdysteroid uridine 5' diphosphate glucosyltransferase. It inactivates the 20-hydroxyecdysone by putting a sugar from a donor onto the hormone.
It's not clear to me why they would hypothesize that would induce this climbing behavior. Is that?
I didn't quite get that either, but I think the thinking is something like maybe at the normal stage in the life cycle of these caterpillars when they're going to pupate, this is induced. That's a guess.
Uh-huh.
And somehow the virus is capitalizing on that and trying to get them to climb up, thinking that they're pupating. I'm not sure. But somehow they hit on the idea that this gene might be involved.
If nothing else, this is a gene that is tinkering with a caterpillar hormone.
Yes.
Yeah. So, I mean, they think that basically when the hormone goes up, these guys would molt, but if you reduce the level of hormone by inactivating it, there's no molting, and they just freeze where they are up in the tree, and they never come back down again.
Right.
And they die.
Okay, so that's an interesting idea. Okay.
Oh, yeah.
They have a little bit of data in the supplemental material. It's called Basis for Hypothesis. Inactivation of the end of the hormone disrupts hormonal regulation of molting and pupation, stopping the period of feeding arrest prior to every molt. Once EGT titers, which is that viral gene, become high enough in virus-infected larvae to inactivate the hormone, the molting cycle is disrupted, inducing larvae to remain up in the tree in a feeding state rather than crawling down.
Got it.
So it's not like it makes them crawl up the tree.
No, they're up there anyway.
Crawling down.
It turns off the crawling down signal.
Okay.
Exactly. Yeah, they're up there to feed.
They're up there to eat the leaves.
Yeah, they go up at night, they feed, and they come down in the day so they don't get eaten by birds. And
that's interesting because the way the data are presented, it looks as if it prevents them from crawling up. But you can think about it either way.
Yeah,
it's actually preventing them from crawling down. Okay.
Exactly.
Right. That makes sense.
So they have this viral gene, EGT, and what can you do? You just delete it, or you put something in it to disrupt it. So they made baculoviruses where they disrupted the gene with either beta-gal, a lac-C gene, or transferrin, just so they no longer make the EGT. And they plaque purify these viruses, which you can do with baculoviruses. And then they injected them into the larvae and put them in tall plastic bottles and measure how high up they crawl.
I love it.
And they have a single graph here with the kind of virus and the height of the gypsy moth caterpillars. And you can see that when you have the wild-type virus, they crawl high up. You disrupt the EGT gene, and they don't go up as far, right?
Yep.
Or they don't go, as you said, Rich, it could be that they don't go back down. Yeah, that's a good question. So let's see how they measured this. They put them in a bottle, and it's lined with a fiberglass screen so the larvae could climb up the sides. Okay, because the bottle is slick, right?
Right, and then they've infected with the virus.
Is he going again?
Yeah, it sounded like it.
Hello.
Okay, you're
there you
back.
go.
Oh gosh, I'm having a bad Skype day.
Okay.
Sound like a bad air day.
Yeah.
Yeah, so the measuring the height is basically a measure of how high up they go. And
Yeah, so they stick them in these bottles.
Yeah.
And they say heights at death.
Right.
Or
The virus
compared by virus kills them no matter what.
Yeah,
Right.
Yeah.
Okay. So they must be distributed all over the bottle, and the height there they show is some sort of average.
Right.
Okay,
It's
so
an average, exactly.
So that still could. And they don't say how long they left them there. Okay, so presumably long enough so that they could crawl up and down a couple of times.
Well, I guess as long as it takes for the virus to kill them.
Right. Okay, I'm thinking that they have this diurnal thing, right, where they go up during the day and down at night. Okay,
right.
So if we're measuring down behavior, we got to let them go up and go down.
Right.
At any rate,
they don't actually tell you how long.
No, they don't really tell you.
So here it's interesting when they say larvae infected with wild-type virus or deletion strains behave similarly during early and middle phases. During the late stage of infection, the deletion-infected insects returned to the container bottoms and died there, whereas wild-type infected larvae stayed in elevated positions and died. So that's reproducing the phenotype.
Yeah. Okay.
So there you go. It's a single gene that controls this.
Yep.
So that is a gene which controls an extended phenotype. The gene in the virus has an effect on the caterpillar. And I think it's so cool that this came out just after we talked about this.
Right.
Yes.
It's in September 9th. So it's cool. I love it.
So you looked up this definition of an extended phenotype.
Right.
Somebody did.
Yep. By Richard
What's
Dawkins.
that?
So What is the extended phenotype?
Yeah, definition.
Genes that can influence events well beyond the bodies that they live in. That's Richard Dawkins.
All right, and there were some examples here.
Oh, yeah, there's other examples as well. Actually, there's some interesting ones. In fact, in this article itself, there's an example. A fungus-infected ant is manipulated by the fungus into a zombie, inducing the ants to position themselves in the canopy where the environment is ideal for sporulation and dispersal, and they're held there by clamping down onto a leaf by the fungus, I think.
Yep.
So that's one example.
Cool.
And this is neat because they have a gene that does it anyway.
Right.
Yep.
And
Excellent.
We have a couple of links to interesting stories here that relate to this, one in NPR and one by Ed Yong in his blog. He calls it Liquefying Virus Uses One Gene to Make Caterpillars Climb to Their Doom.
Doom, yes.
Nature is tough. Okay, anything else, gentlemen, before we go on that? That was straightforward, right?
Yeah.
Yep.
Can you think of a way that the results might be misleading?
No.
I can't.
Part of it is I want to believe this.
Maybe it's
no.
not the hormone. Maybe it's something else that's just going along with the hormone, right?
But now that you point out to me, it hadn't really fully registered. I'm still hung up on up and down here. Okay.
Okay.
But now that you point out their description of this, it really is the down behavior that is affected, because the guys infected with the deleted viruses return to the bottom of the container, and the guys with the wild type viruses stay at the top.
Right.
Interesting.
Yep.
All right. Before we go on to some emails, let's acknowledge the support of our second sponsor for this episode, Wiley Blackwell, the leading scientific publisher of books, scholarly journals, major reference works, and databases. Up to the end of October, they're giving us 25% off of all microbiology and virology books, actually giving everyone, not just us. To take advantage of this, go to wiley.com/go/microbeworld. That's wiley.com/go/microbeworld, and you will see their collection of microbiology and virology books with 25% off. We thank Wiley Blackwell for their support of TWiV. Our first email is from Attila, and I forgot how to pronounce his name, which we did get right at ASV. Do you remember, Rich?
He doesn't mind if we call him Hun, right?
Thanks, Alan.
I'm sorry, Attila. You were probably sick and tired of that joke. But.
Yeah. Well, we asked someone in the audience, and they told us we had it right.
And I forget what the answer was.
So, and he says that we thank you for the right pronunciation of my name at ASV, but now I don't remember. Anyway, he writes, Dear TWivers, I have recently heard that some types of herpes virus may protect us from bacterial infections. This made me remember myxoma virus, viral oncotherapy, and how tumor cells have a compromised immune response. Do you think it is possible that one of those long-term herpes infections that cause no harm to the host could protect us from some kind of skin cancer? So I thought of this herpes latency conferring protection against listeria, which we have mentioned several times. But I asked David Knipe, who's a herpes virologist, and he said Skip Virgin had a Nature paper where they showed that murine herpes virus 68 could induce cross-presentation against bacterial infections. There is an old literature on herpes viruses inducing Th1 helper and polyclonal B-cell expansion. That is probably also a major component of the oncolytic virus effects, induction of innate responses and Th1 helper cell responses and inflammation. There could be some protective effect of latent and reactivating infections against other infections and cancer. So far, it has been too much of a long shot for any clinicians or epidemiologists to do any studies. I think that is why we have not eliminated herpes viruses.
So David leaves the door open.
Yep.
Yeah.
Basically, good question, Attila.
It's a good question, yep. Next one is from Bob, who wrote, Manchester United lost the services of Darren Fletcher with an unspecified stomach virus for 4 months. So Manchester United, I take it, is a soccer team?
Yep. Football.
Football.
Vincent,
Right.
Football.
As far as we know, no soccer club sweeps their training and match pitches for fecal and other contamination. So a pitch is a field, right?
Right.
These surfaces are regularly mown, but it would seem reasonable that this would only break up contaminations and spread them wider. Given that some of these players are earning £250,000 a week and may cost up to £50 million to transfer in, would you agree that screening was a worthwhile exercise? And if so, how often should it be done? So I said that it would probably be more likely that Mr. Fletcher got his infection traveling, encountering other people, which they do a lot of. And he said, well, what about urinating or animal droppings on the field? Could that be a problem? And then at this time, I got an email from Dixon from SIDRAP, zoonotic rotavirus transmission in Denmark. So a animal rotavirus may have infected people, 2 people in this case, cow and goat origin. So I don't know. You could imagine that animals can defecate on the fields and that could infect people, but I think it would be pretty rare.
Yeah.
What do you guys think?
I don't. Well, first of all, he's talking about screening. Who are we going to screen? Are we going to screen the field, or are we going to screen the players?
Yeah, and secondly, I not being a soccer fan myself, I don't know if it's customary to defecate and urinate on the field before a game. And perhaps it is. It's whatever the tradition is. But my understanding is this is just from the numbers he quotes, this is a big money sport, and these are going to be professionally maintained grounds. It seems a little unlikely to me that there's a lot of disease transmission going on the playing field.
No, it makes much more sense that it's travel or something like that.
Yeah, and these players, they spend the minority of their time on the playing field. They actually spend most of their time doing the types of things that most of us do. They eat and they go visit other places and they hang out with their friends and travel and so this is most likely some infection, a norovirus or whatever that Mr. Fletcher picked up on some trip somewhere.
How would you disinfect the grass field anyway?
I don't know if that's possible.
Forget it.
You might kill the grass, right? Unless it's synthetic stuff, right?
Exactly. Unless it's astroturf. Unless if you put something on the grass that's going to kill all the pathogens, it would certainly kill all the grass.
Yeah.
And I don't think
there's no point in screening the players, okay? Because rotavirus infections are over in 48 hours.
Yeah.
Okay. Well, but as we've discussed before, in with other viruses, by the time you can detect it, it's too late.
Yeah.
It's an interesting question, though, right?
Mm-hmm.
It's very good. Thanks, Bob, for that kind of a new.
And it only takes like 10 particles of rotavirus to give you an infection. All right? So just a little bit will do it. So you'd have to catch all of the grass. Yeah. Right. And grind it up and probably flocculate it.
Yeah. Yes.
Deep sequence it, give it to Pippas.
So I think the point here is that screening or decontamination is just not practical.
Yeah. Right.
So, but anyway, good point.
Yeah.
The next one is from Jane, who writes, XMRV has come to mean the VP62 clone that was created in Silverman's lab as expressed by the 22RV1 cell line, which has never been in the WPI labs. Okay, Jane, this is not correct. VP62, in fact, was sent to WPI in 2008, I believe. And that is absolutely true. So they did have the clone. So the point here is that the contamination of the PCR samples, which we talked about 2 episodes ago,
right,
is the 2009 science paper showing presence of XMRV in samples from CFS patients. That was a contamination with this VP62 plasmid. Bob Silverman doesn't know where they were contaminated. He had the plasmid and WPI had the plasmid, so it could have been either one.
The cell line may never have been a WPI.
Yeah, that I don't know.
That doesn't mean that the plasmid wasn't
Right, the
there.
plasmid is certainly there.
And that virus grows on, likes to grow on a variety of cells, if I understand
Yeah,
correctly.
that virus is I would say it's safe to presume that it's in any lab that has cell cultures.
It particularly likes prostate cells, but so if they're around, and I know they did have LN cap cells at WPI because they're in the science paper.
Right.
All right, Lombardi et al. Found GAG and envelope sequences 99% similar to the prostate tumor-associated strains of XMRV, and these are VP62, VP35, and VP42. Those are numbers that came out of the original DeRisi prostate study. Yet Silverman's retraction was only for VP62, which incidentally is the clone used as a positive sample in just about all papers that failed to find any wild human gamma retroviruses at all. I wonder if all the conflicting results have something to do with the semantics surrounding the way the labels are applied. Well, XMRV is this specific virus.
Right. There is no distinction.
All of the published sequences and all the database sequences are this same virus.
Right.
Matter of fact, one of the things that originally made people suspicious of contamination is that if there were a genuine circulating XMRV, you would expect that thing to be significantly more divergent from this clone VP62 than what people are seeing.
And in fact, if you passage XMRV in cells, so one theory to get around that would be, well, maybe it's unique among gamma retroviruses in not mutating. But if you passage it for a single round in cells, you see normal retroviral diversity. You see lots and lots of different sequences.
Yeah. If anything, the semantics, pardon me, are being used by individuals who don't want to let go of this to confuse the issue.
Yes.
Okay?
I mean, the problem here is that XMRV is a xenotropic retrovirus, and there are lots of those, right? Mm-hmm. So it seems to me that we're saying there's no XMRV. Whether or not there are other xenotropic viruses, we don't know. There's no data one way or the other. Personally, I'd be surprised if a contaminant such as XMRV turned out to point you to a xenotropic virus that caused a disease.
Right.
I think the likelihood of that happening is right. Vanishingly small.
No, contaminants are not likely to point you in a productive direction.
Exactly.
Right.
And she goes on, when you bring in the years of research on what MLVs do in animal models, when you bring in the several earlier discoveries of retroviruses in patients with ME, called CFS since 1988, when you know people who have at the least improved their illness experience using antiretrovirals in early days, yet you might not be so quick to dismiss the hypothesis. Jane, there is no evidence that any retrovirus is causing ME or CFS. That's why it has been dismissed by most virologists, because, as we've talked about in this show, there's simply no evidence for this.
There was a report, about 20 years ago or so, that we discussed some time ago, that was a report suggesting retroviruses. Was it associated with CFS or was it something else?
Yeah, with CFS it was HTLV, and that's been...
And basically, that's nobody's been able to confirm that.
No.
Yeah, and you can't cite earlier false leads in order to point out why this one's not a false lead.
Exactly. Yeah.
That's illogical.
And anecdotal reference to people improving on antiretrovirals. You're going to get that with almost anything. That kind of thing requires a properly controlled trial, which at this point, given the mounting evidence that retroviruses are not involved, I wouldn't certainly recommend that kind of trial because there are downsides to taking antiretrovirals.
Yeah, and the problem with anecdotal trials like this that are unblinded and uncontrolled is you could come to any conclusion at all just by pointing, because you're going to preferentially point to the positive examples if you believe the hypothesis. So you could come up with, you could get a result like, whiskey cures prostate cancer because some guy who had it drank and then it got better. There are conditions, most conditions in humans will at some non-zero rate improve. And that may be rare, but somebody goes on antiretrovirals and maybe they feel like they improved a little bit. It's entirely qualitative and it's not a data point.
To get to the blood working group 3 results, the following criticisms of the methodology have been made by scientists, patients, and advocates. 1, the samples used were from patients who were continuing with their medication regimes to include ampligen and antivirals. No preservative was used to preserve PBMCs in the samples. Negative controls were not proven to be negative. Spike positives were spiked with VP62. Sample size was too small to draw meaningful results.
Welcome aboard the RMS Titanic. The problem is not that that deck chair is over there and that those 2 chairs are stacked next to that table over there. The problem is that there's a 300-foot gash in the hull because you hit an iceberg. So pointing to individual aspects of this and saying, well, the problem is that the spiked positives were spiked with VP62. Well, yeah, but the real problem is that the lab at the WPI and Rossetti's lab can't tell positive from negative. And you don't get around that by saying what the positive controls were spiked with, just as an example.
Yeah.
The bottom line is that the test that was being marketed and sold to patients is bogus. But definitively, that's what the blood working group Part 3 results proved.
Yeah, I think that none of these impact on the findings, Jane, of the blood working group. That, as Alan said, the virus could not be reproducibly detected by those 2 groups, and no one else detected them.
Even in triplicate samples from the same patient, they would get different results. So that spikes it.
I don't think the sample size was too small. I mean, they picked the original population shown to be XMRV positive. And
right.
I think then your point that negatives were not proven to be negative, well, they were. Yeah, the problem was that only the CFS samples were contaminated. Well, this is the Silverman study anyway. It's different. So
right.
Anyway, as you know, Jane, this is swirling now even further with accusations of fraud and mistakes and all sorts of things. And who knows where it will sort out. But at the moment, there's just no evidence that XMRV or any gamma retrovirus is causing any human disease.
And there's positive evidence that XMRV is not causing human disease.
Yeah, right. XMRV.
It's not just that there's no evidence in favor of it. There's pretty clear evidence against it.
XMRV certainly does not because it's a lab contaminant.
It's a lab contaminant.
Yeah. And nobody on this podcast has any axe to grind.
No,
right?
No,
we're
we
just
don't.
following the science.
And you know, I would have loved it if this turned out to be a new human retrovirus. That would have been an awesome story.
Yeah.
Sure.
But that's not how it ended.
All right, so the next 2 emails I put in, well, we've been taking a lot of heat on our podcast, so I thought I would put these in. The first is from Conrad, who writes, as a person with ME/CFS, I want to thank you for volunteering to work on the Science Advisory Board. This is of the CFIDS Association. I was touched, though not surprised, by your comments on this week's TWiV about meeting ME/CFS patients. You had kind of a baptism of fire in your encounters with ME/CFS patients reacting to your TWiVs about XMRV. Some people would have turned their backs on both the disease and the patients after that, or decided that we are all indeed just a bunch of crazies. The fact that you haven't speaks to your humanity and empathy. Somehow you are able to understand how someone who is desperately ill could lose their objectivity, and you never seem to assume that the ones who are angry and unkind speak for all of us. You also haven't gloated or seemed happy to learn that XMRV wasn't proved to be the cause. We need virologists of your caliber working to solve the puzzle of this disease, but even more, we need people with your compassion, so thank you.
Nice letter.
Yes,
well put.
Thank you.
Nice letter.
And then there's one, there were some comments from the online version of TWiV. Thank you for your continued coverage of CFS and taking time to help the CFIDS Association. I cannot thank you enough for this. As a patient with CFS, I would love to see an episode dedicated to CFS, as you mentioned. Logical scientific discussion with patients and treating physicians would really bring it home to people that this is real and serious. The outlandish comments seen in news and blog articles have really overpowered a lot of rational dialogue on CFS, and I worry it is doing a great deal of damage to the disease's reputation. This has only amplified with the recent mess of XMRV. I'm hoping an episode on CFS could help clarify to the people we need the most, the scientific community, that the rambling comments they see online are a gross misrepresentation of the patient population. And from another comment. Thanks for sticking with CFS and being part of the CFIDS Association Science Advisory Panel. The ME-CFS community has, I believe, benefited greatly from your objective inputs on XMRV, and good luck with your work with the CAA. And finally, we had suggested interviewing some MD, PhD, people with MDs and PhDs. And so we had 3 suggestions from 3 individuals: Jeff Taubenberger, Jose Montoya, and Patrick Moore. All good people. So
Good. Great.
We'll try to do that. Should we read this last one?
Yeah, because Brian's?
Yeah.
Yeah, because this brings up an interesting thing that is worth discussing.
Okay, Brian writes, at the beginning of the podcast, you say that's TWiV 150, I guess, or 151. You say most of the CFS patients you talked to could remember the day their problems started with an infection. That would be 151. There are many other syndromes that also begin with an infection. For one example, Guillain-Barré syndrome, which can result in paralysis. I think your answer is correct that CFS or ME-CFS can be the result of an unusual immune reaction to any pathogen. It is much more likely to be an uncommon response to a common pathogen than the typical response to an uncommon pathogen. It is also quite unlikely that there is a common precursor pathogen like XMRV that then requires a trigger to set off CFS. I'm a PhD virologist, not an MD or immunologist, but it is my understanding that Guillain-Barré syndrome is detectable not only by the symptoms, but also by being able to measure the human antibodies that attack myelin in each patient. There are dozens of other illnesses, syndromes that are also caused by somewhat unusual or uncontrolled immune reactions to common things. Allergies are one. Chronic inflammatory demyelinating polyradiculoneuropathy, CIDP, is another. In many of these, there is no line in the sand between a severe case of CIDP and a mild case of Guillain-Barré, for example. They are all related and sometimes perhaps even identical in every detail except the level of reaction problem. It's very likely that some cases of CFS are undiagnosed or misdiagnosed Guillain-Barré or CIDP. Other cases may be lingering or permanent effects of an EBV infection. Some percentage of the cases are sure to be not immune-based but depression or other causes, just as there are hundreds of ways to end up with paralysis symptoms somewhat like Guillain-Barré symptoms. Many other causes of chronic fatigue, such as heart and lung dysfunction, get accurately diagnosed so the patients do not end up with an ME-CFS label on their problem. It seems to me that many of the problems with CFS have to do with lack of insurance coverage, fraudulent doctors, treatments, poor patient understanding of the fuzzy lines between one syndrome and another, etc. The XMRV story was quite a fiasco, and it is not over yet. I hope the Science Journal and USA Medical Science can learn something from this. This is from Brian, who is working at HIV databases.
I like the way Brian thinks.
It's very good.
Yeah.
So I was thinking about along this line after our last show, because we were talking about Ian Lipkin potentially doing deep sequencing on all of his samples. And if this is a sort of a hit and run type of disease, he won't necessarily find anything. Okay.
Yeah.
It's one of the models for autoimmunity is that what happens is that you encounter some sort of an antigen, like say through an infection or something like that, and you mount an immune response to that, and then because of some sort of subtle malfunction in your immune response, that immune response is cross-reactive to some self-antigen, say myelin basic protein in your nerves. The classic is the mouse model where you shoot them up with myelin basic protein and they get something like multiple sclerosis because they mount an immune response to their own myelin.
The EAE mouse.
Yeah, this is still a very popular model of autoimmunity. So it could be that if ME-CFS has an autoimmunity component that, as he says here, you might have an encounter with a pathogen and not necessarily a unique pathogen that triggers this thing, but then it's gone. And years down the road, you may not find some sort of a pathogen associated with a patient because that was long in the past. What it did was trigger a response that causes a continuing problem.
You know, so in that case, you need to have patients early on in the disease, right?
Right.
If you can get them. So
right.
People who first, but sometimes it takes a long time for them to realize that they have this. So that's hard because, as you say, the agent may be gone by then.
And then go ahead, Alan.
I kind of think that there's a very high probability this is going to turn out to have complex causes.
Mm-hmm.
Yeah, I agree.
That it's an infection and an immune response gone awry and other factors, and there's just a whole lot of stuff that can happen to people out in the world, and this set of symptoms that is called CFS, or whatever name you apply to it, as Brian points out, there have got to be multiple pathways by which you can end up at such a nebulous endpoint, because it's really, even no matter which set of standards you look at, there is a lot of murkiness in this diagnosis, and I'm certain that there are people who've ended up in this bucket in different ways, that there's not one mechanism behind it.
Well, the most important thing about all of this that we've been through is that, as I said before, at least in my mind, it's raised a consciousness of this disease, and I hope that it's had a similar effect on a broader scale. I don't know that, but I hope so that some more appropriate attention can be paid to it.
Sure.
All right, thank you, Brian, and let us move on to some picks of the week. Rich, what do you have?
This seems to be a Steve Jobs tribute show down here at the end.
Yes.
Everybody's talking about Steve Jobs. Somebody pointed out to me the 1984 Apple Macintosh commercial, which I guess I had seen, but it didn't stick in my head the same as some other people. So when the Apple Macintosh first came out in 1984, they made an advertisement for it that ran just once on the Super Bowl. And there are those, and it was directed by Ridley Scott.
Yes.
And it was, it has been touted by some as the most amazing, most effective commercial ever made. And it's got a scene out of George Orwell's 1984, and the theme basically is that the Macintosh is going to break the mold. It's going to be a revolution. And 1984 will not be Orwellian because of the Macintosh, right? So I looked this up this morning. I had a look at it, and it really is pretty impressive. And it's a nice little tribute to Steve.
Yep.
Yeah.
Alan.
Well, I noticed you guys had done this theme, so I decided I would stick with it. And I just today I read this XKCD webcomic, which has been a pick of mine before, but today it's this tribute to Steve Jobs, and it's I thought it was quite clever. I thought it was really brilliantly done. So the two characters are sitting there on a wall looking out at this little memorial garden with the eternal flame, only instead of a flame coming out of it, it's the spinning beach ball that you get on a Mac when some process has gotten hung up. And if you're not a regular XKCD reader, you may not know that if you hold your mouse over the comic, you get a little pop-up mouse over text. So it gives you an additional punchline to it. So you hold it over with this one, and the mouse over text is, there's always the hope that if you sit and watch for long enough, the beach ball will vanish and the thing it interrupted will return.
I can't tell you how many times I do that.
Yes.
Just waiting for it to go away. It's great.
Yeah.
That's really nice. I had another pick, and last night I saw Rich had put the commercial on. So my pick is a video of Steve Jobs' 2005 commencement address that he gave at Stanford. And he had just come back from cancer surgery, and he was talking a lot about death. It's a really interesting address. One of the parts from it goes, "Your time is limited, so don't waste it living someone else's life. Don't be trapped by dogma, which is living with the results of other people's thinking. Don't let the noise of others' opinions drown out your own inner voice. And most important, have the courage to follow your heart and intuition. They somehow already know what you truly want to become. Everything else is secondary." Anyway, the whole speech is really good. He gets very personal, which he didn't always used to do, and there are a lot of lessons to be learned. So there you go, a little mini tribute.
My sense is that's
Jobs.
the way he lived his life as well.
Yep. Yeah, it's very interesting to see the people's comments all over the place. You know, Obama, Mark, what's his name from Facebook?
Zuckerberg.
Mark Zuckerberg. All these CEOs of various sorts, Bill Gates, et cetera. It's very, very interesting. Lots of stories everywhere. Apparently there's going to be an entire Time issue. They've canned their normal issue this week to do a special Time issue on him. So
56 years old.
Yeah, that's the thing. He's younger than me.
Younger than me.
Wow.
He's not a whole lot older than me. Yeah.
I can go anytime. So enjoy your life.
Right.
There you go.
Yeah, he said, part of that Stanford speech is he says he looks in the mirror every morning and asks, if this were my last day, is what I'm going to do what I would do? And if the answer is no, too many days in a row, it's time to do something different.
Yeah, I mean, that's tough because I spend days wasting too much time.
It's hard. Well, if nothing else, if you're going to do it anyway, if you sit there saying, I don't want to do it, then you really are wasting your time. If nothing else, you can rewire your attitude. Yeah,
I end up starting out a day with good intentions, but then there's a lot of wasted stuff, you know. I don't know. What am I supposed to do? Anyway, it's a good sentiment. We also have a listener pick of the week from David, who said, a suggestion for a pick of the week, the book Your Inner Fish, A Journey into the 3.5 Billion Year History of the Human Body by Neil Shubin. I like this book because it's a scientific journey. Shubin starts with a hypothesis. Given our understanding of evolution and geologic time, we should be able to find a water-to-land transition species preserved as a fossil in a certain age of rocks of a certain ecology setting. Shubin details the expedition to a Canadian Arctic site of the right age and environment to find such an animal, eventually finding Tiktaalik, a fish with distinctly alligator look to it. Shubin also covers how various body parts have evolved, hence the title From Our Fish Ancestors, a great read.
Cool.
You guys have not read that before, I presume.
No.
Neither have I, so thank you for that, David. And that'll do it for TWiV 152. You can find us at iTunes, the Zune Marketplace, also at microbeworld.org and twiv.tv. Don't forget to send us your questions and comments to twiv at twiv.tv. Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
You're quite welcome.
Always,
it's a gas.
Always a gas. And Alan Dove can be found at alandove.com. Thank you, Alan.
Always a pleasure.
And Vincent Racaniello at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.